All right, so thanks again for joining us. I am Mirta Truba, sales engineer at ASI. ASI produces hybrid pixel detectors based on TimePix and MEDIPIX technology developed by CERNs. Lots of scientists have benefited from this technology in many fields and applications, including BMI, quantum optics, atom probe tomography, and many more that you're very welcome to check on our website. If you have any questions during the online seminar, please leave them at the chat, and either our moderators will pick them up or we will address them at the end. I am very happy to introduce our guest speakers today, Professor Thomas Weinacht and researcher and PhD candidate Chuan Cheng. Dr. Thomas is a physics professor at Stony Brook University. His research is focused on time-resolved spectroscopy and quantum control. He's a fellow at the American Physical Society and the author of the book Time-Resolved Spectroscopy: An Experimental Perspective, which is a concise guide to the basics of time-resolved spectroscopy. Researcher Chuan, also from Stony Brook, focuses on strong field science and momentum imaging of ions and electrons in coincidence. We are very excited to have them here today to learn more about their experience working with TPX3CAM, measuring the 3D momentum of ions and electrons. The platform is yours.
Okay, great. So hello and welcome everybody, and thanks to ASI for organizing this seminar. So I'm going to share my screen in a moment, and the experience is that when I do share the screen, I can't see the questions, but you can ask the questions and Mitra will forward them to me, and I will happily address any sort of clarification questions that people have along the way. And then, of course, we can have a more involved discussion towards the end for larger questions. But I'm sort of assuming that this is perhaps a broad audience, and so there may be things that are specific to the science we're going to be talking about with the camera that bear some quick clarification. So I'm going to start sharing my screen, and I hope that everybody can now see the PowerPoint presentation. I'm going to go to full screen presentation mode. I can't see anything from the presentation. I don't know, is my screen visible? And okay. Okay, great, thank you. Okay, so I'm going to continue then. So what Chuan and I are going to talk about today is basically the application of the TimePix camera for 3D momentum imaging of ions and electrons. And we're going to focus on experiments in our laboratory, which are on sort of time-resolved spectroscopy and strong field laser matter interaction. Here's a cartoon diagram of how we make use of the TimePix camera to image ions and electrons. And here's real life. So you can see the TimePix camera is right here, and a velocity map imaging apparatus that we have is below, and there's a mirror here which is just reflecting the light from a phosphor screen to the camera. And I'll talk a little bit about how, first of all, why are we interested in what we're doing, and then how the TimePix camera facilitates the kinds of science that we're doing. And Chuan will go a little bit more into detail about how we convert the timing and position information from the camera into three-dimensional momentum information about the ions and electrons. So a brief motivation comes from sort of, a long time ago with the original sort of question of Muybridge and the horse, which is that do all four legs of the horse leave the ground at the same time? And that was not something that people could resolve by looking at the horse running because the image was a blur. So the exposure time of your eye to the images that you were looking at were too long. And so a single frame, so to speak, captured by your eye and your brain was not short enough to resolve the motion of the horse. And that was, in a sense, the beginning, one could say, of the idea that we would like to take very short exposures of rapid dynamics in order to try and put together a movie of sorts. And what I'm going to be talking about is essentially making a molecular version of this, where we'd like to take snapshots the kind of movie that's shown here, which is the molecule CH2I2 breaking apart after photoexcitation. And here you see a beautiful movie created by Philipp Markotand from the University of Vienna, which is illustrating what happens for a particular excitation process when we drive this with UV light. And what we're interested in doing is using time-resolved spectroscopy to capture those dynamics. So we're interested in making a film and performing pump probe measurements where a pump pulse will initiate some sort of dynamics, and a probe pulse captures those dynamics, taking a fast picture like the bullet entering and exiting the apple here. The picture is something that we need to be a little bit more creative in interpreting. And similar to the way that you have an MRI of something in your knee, we need to reimagine what it means to take a picture, because clearly the objects that we're trying to take a picture of are below the diffraction limit. And so we can't sort of take a picture in a conventional sense. And the way in which we are interested in doing the picture taking or the imaging is to look at the electrons and the ions that are emitted from the object in question, the molecule that's undergoing some sort of dynamics, and imaging those ions and electrons in order to maximize the information content that we get from them. So that will allow us, as I'll hope to describe in the remainder of the talk, to not only get a sort of a structural view of the dynamics, which might be facilitated by other measurements such as diffractive measurements with X-rays or electrons, but also an energetic picture of how the energy of the molecule might evolve as a function of time, and how non-adiabatic dynamics such as internal conversion play an important role in the evolution of the molecule. So the question is, how does imaging ions and electrons play into this goal of following molecular dynamics? Well, the idea is that if we, in an energetic picture, prepare a wave packet in an excited state by absorbing light from a pump pulse, such as this blue one here, then we can prepare an excited state wave packet which is evolving on the excited state potential energy surface. And then by having a second probe pulse ionize the molecule, we can then have an observable, which can be the ion or the electron produced by that ionization. And we can now study how that ionization yield depends on the delay between the two pulses. The issue is that we need to measure those electrons and those ions. And the more information we get from the ions or the electrons, the more information we can glean about the intervening dynamics that was following the pump pulse. For instance, you can imagine that as the wave packet rides down on this potential energy surface here, the energy available to the photoelectron with the absorption of a secondary probe photon will decrease with time delay. And so if we measure not only the yield of the electrons, but their energies as well, then we can get more information about the intermediate dynamics. So the question is, how do we measure those electrons and those ions? And we make use of velocity map imaging. And the idea here is that if we have a molecular beam that produces a source of our molecules of interest, and we intersect those with a laser beam, then we produce electrons and ions at the sort of this point in this ion and electron imaging system. And what this imaging system does with the appropriate geometry and voltages applied to these metal plates here is map the momentum of the emitted particles to position on a detector here. And what I'm showing you here is sort of traditionally what has been done is, in a sense, two-dimensional velocity map imaging, where the X and Y, or in this case, the Y and Z momentum of the particle is mapped to the YZ plane here of this two-dimensional detector. And then if you have a three-dimensional distribution which has some cylindrical symmetry about, for instance, the laser polarization vector, then one can use an inverse Abel transformation to reconstruct the full three-dimensional distribution. But that is sort of a little bit less desirable because it requires acylindrical symmetry, which can be violated if the polarization is not linear. Like with elliptical polarization, we would violate that. And also, it requires, it precludes certain kinds of measurements, like coincidence measurements, where we measure ions and electrons together at the same time. So what would be lovely is to measure the full three-dimensional distribution, but that's difficult with a two-dimensional detector. And in a sense, that's what this talk is about, is the fact that the TimePix camera acts essentially as a three-dimensional detector, because not only do we get X and Y information in this plane here, but we also get timing information, and that timing information facilitates the full three-dimensional measurement. And that's sort of illustrated in this next slide. And after this slide, I'm going to pass the stage to Chuan, who will tell you a little bit more about the details of how this camera operates and how that facilitates the measurements. And then I'll come back at the end of the talk and say a few words about how we sort of do some of the other time result spectroscopy with the camera. But the basic idea is this, is here's the velocity map imaging that I described earlier. Particles come and hit our detector here, which consists of a microchannel plate, which basically changes one electron or ion into something like 10 to the 6 electrons. And then those 10 to the 6 electrons are incident on a phosphor screen, which emits light. And then that light is imaged to the sensor of the TimePix camera. And because the TimePix camera not only records the two-dimensional position of each electron or ion arrival, but it also records the time with a precision or a resolution of about a nanosecond. It allows, for instance, here, this is a source of ions that Chuan measured in the lab, where we not only get the X and Y information, but we also get the longitudinal information, because the time at which an ion hits can be converted into the longitudinal momentum. And so that's what this sort of talk is really about, is that we can use the TimePix camera to measure three-dimensional distributions of ions and electrons. And the first part of the talk will sort of focus on how we can do this for 3 dimensions with ions, 2 dimension for electrons, and then we're going to sort of touch on how we can do this 3-dimensionally for electrons in the end. But it's a little bit trickier because the electron arrival times are much more closely spaced, and so the timing precision that one requires is much higher. So at this juncture, I think I'm going to stop sharing my screen. I'm more than happy to try and address any questions that might have arisen in this first few minutes. And I'll pass the sort of stage over to Chuan, who will talk for something like 20 minutes, and then I'll come back at the end. So if there aren't any immediate questions, then I don't know, Chuan, do you want to take over and share your screen?
Sure, I can do that.
Okay, so there was one quick question here, which I'll try and address, which is how does the multi-channel plate affect the timing? So I'll give a brief answer here and then come back to that a little bit later. Indeed, one has to worry a little bit about a number of little issues with the microchannel plates, particularly for the detection of electrons. So for ions, the microchannel plates basically introduce very little delay or variation in the timing with signal height compared to the distribution of times that the ions are spread over. So it's something that you basically don't have to worry about. But in more recent work where we've looked at photoelectrons, then the sort of picosecond timing that we're interested in is important. And I'll talk a little bit some of the technical details with how we handle the electrons and the microchannel plate issues toward the end of the talk. So one more question there was that this is for any ion, also for hydrogen. Essentially the answer is yes. The reason that hydrogen can be a little bit trickier is because we do coincidence detection, and Chuan will discuss this in a little bit, so I'll sort of leave him to answer the question, the bulk of the question, but it works for any ion, and we'll talk about the details, both myself and Chuan, a little bit later. Most of our recent work has focused on deuterium because it's a little bit easier technically in terms of when you switch the voltages on the plates to look at both electrons and ions. But in terms of the camera, I would argue that basically hydrogen is not a problem.
Yeah, I'm going to show this 3D imaging of the ions in a second, so you have a taste of how they look like on the detector in a bit. Okay, so I guess it's time for me to start. So let me share my screen. Share. Okay. Can you see the slides now? right, view start. So, so here I'm going to show you how we use the TMPX3 camera in a real experiment with an example of the, sorry, let me put the laser pointer on. With the example of a strong field ionization of the deuterated water molecule. So, so here I listed the several properties of our laser, which is a 10 femtosecond laser pulse centered at 780 nanometer. And then we focus into the chamber to reach about 400 terawatts per centimeter square. This intensity is strong enough to do single ionization and even double ionization. And once we get ionization events, we first accelerate the electrons to the detector, and then we flip the voltage on the VMI plates to accelerate ions. So in this way, we can get a coincidence event of both electrons and ions. So here I'm showing you the data format of the TMPX3 camera. Essentially, the camera stores data in this 64-bit packet style. And there are different types of data packet. For example, there is this TDC packet, and there's also the pixel packet. Well, the TDC packet has very nice timing resolution. That's about 0.26 nanoseconds. So we use it for our trigger information. And in the real experiment, we are the real-time resolution is this determined by this pixel packet, which is the bulk of our data. So that's a very fast time resolution. And on the other hand, the pixel packet also has this 16 bits saved for the XY information. So this is a very remarkable difference between TMPX3 camera and also and the other commercial cameras, because the commercial cameras usually what they do is they save a lot of pixel informations, i.e. the pixel's voltage or intensity given in a whole frame. Well, for TMPX, only the pixels that go over the voltage threshold that can be saved. So the TMPX camera already reduced the amount of data to be saved by a lot. So on the other hand, the TMPX3 camera also saves the intensity of the pixel indirectly by this quantity called TOT. So it's short for time over threshold. And what it does is pretty self-explaining because think about a voltage of a pixel. So when it gets a flux of photons, it could go above threshold. And the amount of time that it stays above the threshold is this time over threshold. So this makes the TMPX3 camera pretty resistant to the room light because the room light level is usually just way below the threshold. It is when we get a flux of photon, then it starts to crank up pretty fast. But on the other hand, there's something not very great about this mechanism because let's think about, oh, if I have a high flux of photons, then the voltage will climb up faster. But if I have a lower flux of the photons, then the voltage will climb up slower. So that introduced a uncertainty determining, oh, when is my real time of arrival? So we will need this flux versus time of arrival correction. And the flux is in some sense related to the time over threshold so that we can use a time over threshold to make this correction. So here I'm showing you the example of how this TOT correction works. So basically we're looking at this fragment called CH2Br. Well, bromine has two isotopes that is almost of the same abundance in nature. That's why we are seeing two peaks. And so if you look at this 2D histogram from pixels, we can see that there's a clear relationship between the TOT on the y-axis and the TOF on the x-axis. So if we apply centroiding algorithm on them, then the new histogram on centroiding, it's getting way much better, but still it seems to show this kind of correlation, which is not what we want. So we can fit this correlation and then apply the opposite correction. then we finally reach this very straight vertical lines up, and yeah, vertical lines. So at that time, we will have the best time resolution. Okay. So it's important to note that in the real data stream, we are actually it's a little bit complicated because the data is actually increasing with time, meaning that if I have a laser coming at say 1 second from the start of the experiment, then my electron comes in maybe at 4 microseconds, 30 nanoseconds after that, then in the data stream, what it looks like, it's going to save the electrons arrive at not the 30 nanoseconds, but 1 second plus 30 microseconds. So in order to get a meaningful time of flight, we will need to fit in the laser's time by sending a laser synchronized input into the camera through this TDC input. And the TDC input, like I'm showing here, it's going to repeat itself every 1 millisecond, which is exactly our laser repetition rate. And the difference between this time and also the time of arrival gives us the time of flight that we are looking for. Okay, so here I'm going to show you a video. So essentially what this video does is that it's showing, displaying the frames every 100 milliseconds. So in our experiment, this is 100 laser shots per frame. So we can see that the inner part of the image seems to have smaller hits, while the outer parts of the image seems to have bigger hits. Well, this is not actually not because of the bad camera sensitivity, but rather because our MCP, our detector, is aging through time, and because the center of the detector has more hits in the history, so that the efficiency there just gets worse comparing to the outer side. So so that will introduce a luminosity difference between the center and the outer side, and reflecting on the camera, it's the hit size difference. So on the other hand, we can see that the camera is pretty resistant to the room light, as I was talking before, and also we see that these hits are almost on the order of maybe 50 pixels in area. So since we are using a 1 gigabit per second Ethernet cable, so the simple simulation tells that we can actually handle 300 hits per laser shot. Well, on this image, we can maybe see less than 200 hits. So divided by 100 laser shots, that means I'm only looking at 1 or 2 hits per image or per laser shot. So that's way lower than this upper cap of the real ability of the camera. Okay, so so here I'm showing you the time of flight converted to the atomic mass of the fragments of the deteriorated water. And then now let's turn our eyes to how we get the 3D image of the ions. So we get the X-ray from the camera, right? And then we know that the X and Y are proportional to the transverse momentum of the molecule, of the fragment. And then we also know that the time is roughly proportional to the square root of a mass to charge ratio. Then the question is, oh, we know that there is a small deviation that might give us this PZ information, but is that proportional to the PZ, or is that proportional to the PZ square? So a simple simulation seems to show that this deviation to mass to charge ratio, it's pretty linear with respect to the momentum in Z direction. So this effect combining with the XY gives us this full 3D momentum. And so on the right, I'm showing you the deuterium coming from the D2O strong field ionization, and then the reduced dimension polar coordinate. And even further, we can calculate the 1D information, the momentum or the energy. So by now, I guess I have shown you guys some how we do the 3D ion imaging. There's no need for able inversion, and there's no need to do this DC slicing stuff. Okay, great. So for the electrons, the story is a little bit complicated because we do not have enough time resolution, or we do not have enough time span to do this the third dimension. So what we do is we do this able inversion on the XY image of the camera of the electrons, and then it gets us to this polar coordinate. And further on, we integrate over the theta angle to get this 1D histogram with respect to the momentum or the energy. So in this case, for the electron, we are able to do this 2D and with the able inversion, but we cannot do the DC slicing or the 3D. Okay, so since we are doing this experiment in a coincidence mode, that means we can look at the photoelectron spectrum in coincidence with different ions, like I'm showing here. So we can see that the photoelectrons looks pretty periodic, but they look noisy. And so let's just focus on the parent ion and the deuterium. And a little bit more words about the periodic structure. So the for a particle, it can absorb more than enough photons to get ionized, like I'm showing here. So the excessive energy will be mostly distributed to the electrons, and then it's separated by the photon energy of our laser. So this structure, it's periodic structure, is called the above threshold ionization. We usually call it ATI combs, like we can see on the right, which is coming from this xenon atom experiment. On the other hand, there's something interesting about the excited cationic states. So for those states, because their ionization potential is different from the ground cationic state, then what it's going to look like is they're going to look shifted. Their ATI combs will be shifted comparing to the ground cationic state ionization. So for molecules, this is a bit more interesting because it could have different dissociation channels. And the different dissociation channels have different appearance energy and the asymptotic energy. And that might highlight this excited cationic state. And so if you look at the photoelectron spectrum in coincidence with this channel, we'll be able to select this ionization channel. So this is a pretty unique feature for molecular ionization. Okay, so now I'm showing you this photoelectron in coincidence with deuterium and the parent ion, the D12. So we know that for parent ion, the majority of the electrons are coming from the ground cationic state by removing a HOMO electron. Well, HOMO just stands for the highest occupied molecular orbital. So that's the outermost shell. Well, then the question is, which orbital or which state is corresponding to the ionization of deuterium? So it turns out that it is this HOMO -2 state. Well, sorry, it's a HOMO -2 orbital. So that is the second orbital that's below the HOMO. Okay, so now let's have a deeper look into the deuterium. I already showed you this 3D view of the deuterium and this polar coordinate. And in this polar coordinate view, we can see that there's a very strong angular distribution at this high momentum part. So that corresponds to this Coulomb explosion of the double ionization when it has two fragments that have both have positive charge, it's going to Coulomb explode. So if we plot the 1D histogram of the energies, we can see that the peaks are matched sorry, very well with this coincidence of the deuterium and its complementary part, the OD fragment. But there's something here that's missing. So it turns out that those areas are actually referring to the so-called three-body dissociation. So the two-body dissociation just means this deuterium and the OD ion. Well, the three-body dissociation is two deuterium and one oxygen. got departed completely. So we can separate these two channels by looking at the kinetic energy, but also we know that there's angular distribution that we can use as well. So one thing that we would like to try is to look at the photoelectron spectrum in coincidence with these distinctive features. Okay, so here I'm showing you how they look like for the two old ones. So they are very nice. Well, the new ones seems to be kind of complicated and they look noisy, even though they line up with the peaks that we already see. So what do we do? So in order to have a better look, what we did is to compress this ATI orders into one order by just taking slice of each order and then summing them up into the one order. So here is how they look like by the end. So for the two old ones, they seem to be picked at where we want. So for the parent ion, it is picked at this removing a HOMO electron peak. Well, for the HOMO -2 electron, it is picked at this peak. Well, interestingly, for the two-body dissociation, it is picked at this removing HOMO electron instead of having the same one from the single ionization's dissociative channel. So it's consistent with the parent ion. On the other hand, the three-body dissociation seems to have three distinctive peaks. Well, that's also quite interesting. So how could that be the case? Well, it turns out that it's related to which states leads to this two-body dissociation and three-body dissociation. And actually, it is the lower lying states of the dication that tends to do more dissociation into the two-body, and the higher lying states tend to do this three-body dissociation. And so if you check the electronic configuration for those dication states, we find that these lower lying states are highly depending on this 1b1 orbital, which is exactly this HOMO orbital. Well, for the three-body states, so where the states are higher lying states, their state configuration are more complicated. That's why we are seeing three states. So great. We see that for the double ionization, their coincidence electrons show signatures of the intermediate ionization's features. Then the question comes, okay, can we see direct signatures of these double ionization states, not from this intermediate step? So there's a way that we can do it, which is essentially to look at ions. Basically, we have this dissociation channels, right? So they will have this dissociation dynamics going on. So what we could expect is to look at this kinematics of the final fragments, and then hopefully they will show this distinctive features from different states. And so because of a limited amount of time, I'm not going to go to the details, but essentially see that this a very nice single photon double ionization experiments taken. And also this simulation captures these interesting features from it based on this ion-ion constants. And we compared it with our strong field results and read it with a manuscript, which is currently under review. So it is available at this archive link. So please check it out if you're interested. So the above experiment I'm talking about, it's either on electron on ion constants or ion-ion constants. There's no external parameter scans that we did in that experiment. But one can think about ways to do this parameter scans. For example, because the real time of flight is the difference between the TDC information and the pixel information. So the pixel information is always synchronized with the real time of the laser. For example, the electrons will always come at maybe 30 nanoseconds after the laser comes to the chamber. Then well, we can play some games with this TDC information by saying that, oh, I can control my delay generator of the laser so that it can delay my TDC signal by 10 microseconds each time, meaning that for my first trigger, it's the same time together with the laser. Well, for the second trigger, it is 10 microseconds. This time it's earlier than the real laser. And the next one, I want it to be 20 microseconds earlier than the laser. So the time of flight, it's equal to the difference of the two, and then it's going to be masked by the delay index that I have. And that delay index can be synchronized with this kind of palm probe delays. And so that we can use this delay index at any kind of parameter scans that we want. So, okay. I hope we have a good view of how we use a camera. Like I said, this parameter scans, it's utilized in a lot of things. For example, the time resolved photoelectron, photoions, spectroscopy. So now I'm going to give this back to my advisor, Thomas, to discuss further on that. Okay. Sorry. How do I stop sharing?
Okay, I guess, is my screen being shared okay? Chuan, can you see my screen? Okay, great. So maybe what I'm going to do is, in the interest of time, I'm going to sort of continue what Chuan had been talking about a little bit just now, about how we can sort of synchronize the camera data stream with some sort of external parameter that we're scanning. And as I alluded to earlier, we're very much interested in scanning, for instance, a pump probe delay. And so what I'm going to simply illustrate now is how we can use the camera to do pump probe measurements and measure the kinetic energy release of ions. So, you know, there's some time resolved photoelectron spectroscopy results that I was going to talk about briefly, but just to make sure that we finish within the 45 minutes, I'm going to sort of not talk about those in detail, but just give you an example of how, you know, what Chuan has talked about is using the camera for imaging ions and electrons in coincidence to look at strong field molecular ionization results. And here I'm going to just illustrate how, in a sort of a more weak field approach, it can be used to measure, for instance, the kinetic energy release of ions and follow the conversion of electronic potential energy into nuclear kinetic energy. And what's really attractive about the camera is that it allows you to sort of measure all ions at all times with velocity information. So you don't have to slice out with the detector voltages a given class of ion, like a certain fragment. You can see all the ions all the time, and you can get all of their velocities. And so that's extremely attractive. And I'm going to illustrate how we use that to sort of follow the conversion of electronic potential energy into nuclear kinetic energy in a pair of molecules. So here what I'm showing is a diagram, which is a one-dimensional cut of this molecule I had mentioned earlier, CH2I2. With UV excitation, we get a combination of internal conversion between sort of a group of states that's cartooned here as light blue and a group of states cartooned here as red. And then what we're looking at is basically the competition between internal conversion and dissociation. And and we can measure the photoelectrons, and we can also measure the photoions. So what I'm going to describe now is how we use the camera to measure the photoions and then follow the conversion of electronic energy into nuclear energy. And in particular, for the molecules that we're going to be talking about, CH2I2 and CH2IBr, there's sort of three groups of states: one which is bound, one which is dissociative and leads to a spin orbit excited iodine, and one which is dissociative and leads to a ground state iodine in the dissociation process. And so here are the sort of, in some sense, raw measurements in the top left and top right corners. And then in the bottom left and bottom right panels, for the two different molecules, CH2I2 and CH2IBr, you see sort of self-normalized kinetic energy spectra. That is the top left and top right panel, where we just normalize the spectrum to each delay. And then I can show you sort of more easily how the kinetic energy evolves independent of how the amplitude of the peak changes. And what you see is that there is indeed an increase in the kinetic energy release of the particles, as you would expect as the molecule dissociates. Then probing at later time delays allows conversion of energy into nuclear kinetic energy, and that leads to an increase in the kinetic energy release. But what's particularly interesting here is that we see that for CH2I2, we see a single peak here, which is increasing in energy, whereas for CH2IBr, we see two peaks here, and we see a significant difference in the kinetic energy release. Calculations agree exactly with this sort of behavior. So here's theory and experiment for CH2I2, theory and experiment for CH2IBr. And these measurements can essentially be interpreted by examining the calculations in terms of competition between dissociation and non-adiabatic dynamics, that is, going through conical intersections or non-Born-Oppenheimer coupling between electronic potentials. In CH2I2, this happens during the dissociation, so there's lots of hopping between potentials if one thinks about it in a sort of a surface hopping dynamics kind of way, which is the way that the calculations were carried out. Whereas in CH2IBr, all of the internal conversion or non-adiabatic dynamics takes place in the first 20 femtoseconds, and then the wave packet rides out in one of two groups of potentials, and that's why you see the results that you do. So in CH2I2, there's hopping back and forth between different groups of potential states, and that smears out the energy distribution, leading to a single peak, because you don't ride out in a well-defined potential. Whereas for CH2IBr, that's sort of frozen out before the dissociation process. And so there's a very nice sort of resolution of this difference here, which is facilitated by some surface hopping calculations that were carried out by our collaborators, Philip Marketan and Tomasz Rozgoni. What I want to sort of end off on is sort of a very exciting new result that we've had in our laboratory, which is the ability to do three-dimensional imaging for electrons. And I'm going to take the last 2 minutes of the talk to sort of illustrate how we do that and show you some preliminary results that we have. So just to sort of set the stage, if you say, let's imagine that I have two ions. One has 1 eV of kinetic energy and is going up, and one has 1 eV of kinetic energy and is going down. Then the difference in time of arrival for our velocity map imaging apparatus is about 9.3 nanoseconds per square root atomic mass unit of mass. So for two protons that are going up and down with 1 eV of energy, you would still have something like 9 nanoseconds time separation. So with the roughly 1 nanosecond timing resolution of each pixel in the camera, you can still map the time of arrival to the longitudinal velocity distribution and make the measurement. With electrons, it's a little trickier because those same two electrons, if you do the math, given their lighter mass, will arrive only half a nanosecond apart. And with the sort of nominal 1.5 nanosecond timing resolution of the camera for each pixel, it's not possible to convert the time of flight information into longitudinal velocity or momentum. But we have made use of a new feature in a new version of the camera, which is that there are now two TDCs. And those two TDCs both have 1/4 of a nanosecond or a 250 picosecond timing resolution. And so what we do is we take basically we capacitively couple to the phosphor screen of our detector, go through a constant fraction discriminator, and feed that information into TDC number 2. So there's a photodiode, or it's a delay generator that's synchronized with the laser that goes into TDC number 1. And while the photoelectron timing information is not captured on a pixel by pixel basis with sufficient timing resolution to get the time of arrival and momentum of the electrons, we do actually get timing resolution from TDC2 that we can correlate on a hit-by-hit basis with a given group of pixels so that we can get sub-nanosecond timing information, allowing us to produce 3D VMI for electrons that I highlight in this graph here. So a few weeks ago, in collaboration with Carlos Traero from the University of Connecticut, we made some measurements in my lab where we basically looked at above threshold ionization from xenon. And you can see beautifully here, this is sort of, in a sense, the raw data from the camera, where in the Z information here was essentially timing information, and then the X and Y information was pixel information from the camera. And you can see now there's 3 different projections of that information, and we have the full 3-dimensional result here. If we rotate the laser polarization, then you can see the distribution rotates. And now the sort of ATI spectrum is mapped to position of the detector, or we can map it to the time of the detector. And these graphs are sort of aimed at showing you that there's equivalent information whether we map it to time or to space. So in conclusion, this is sort of just an acknowledgement slide telling you about many of the people who have been involved in this work. Andrei Nomorotsky from Brookhaven National Lab was the one who really got us into the TimePix business. And Ganesh Mogul and Chuan were the people who were really most active in the lab in taking and analyzing the data. Chuan has been sort of the resident expert on the TimePix camera in the group for the last few years. And this work was done in collaboration with also Vinu Kunarapan and Daniel Rolas, Phil Buxbaum, Ruri Forbes, Andrew Howard, and other members of my group who are listed here. Yusung Liu took the data on CH2I2 that I showed you, and we've received generous funding from the National Science Foundation and Department of Energy. So that's the end of the slides that I'd prepared, and I'm happy to try and answer any questions that people have on the presentation.
Well, thank you very much, both of you, once again, for this very informative presentation and for sharing your results with us. For our audience, if you're interested in the specifics of TimePix3CAM, you can download the brochure, the product brochure, by going to the handout tab at the top right corner. Keep in mind that ASI is always looking for new projects to collaborate on, so feel free to contact us at info@amsins.com or connect with us via LinkedIn. The recorded version, as said earlier in the chat, will be available and sent to you via email. And now, as Thomas said, let's open the floor to your questions. Our product expert and application scientist, Dr. Jingming Long, will also be joining us, so feel free to ask any product-related questions as well. Hello, Jingming.
Hello, Mirta. Hello, everyone. Yeah, if you have any technical question, please leave your question on the chat section. And of course, I already saw there a question related to experiments. Maybe Thomas and Chuan can help to answer them.
As I can see, there is one question from Xiao Hong. Hi, I have a simple question. How to calculate the momentum resolution from the 2D matrix in TOA, and what's the normal value of the resolution?
Okay, so I guess I can answer that question. So the resolution on our VMI detector is the key point. We are not limited by the camera's resolution. I think that's about what. So for the deuterium, I think our momentum resolution is about 0.2 atomic unit. So, well, it's also related to the voltage that we apply on the VMI plates. So it's really a parameter dependent thing. Well, at that time, I think our time resolution it's maybe a factor 3 of that. So when we convert time to the PZ, then that is about 0.6 atomic unit.
Yeah, just to follow up, I mean, the resolution depends on many parameters, as Chuan indicated. I mean, the tightness of the laser focus plays a certain role. I mean, we have multiple velocity map imaging setups in our lab. And for most of the strong field work, you have the advantage that you focus the laser very tightly. So the source of the ions and electrons is very small. And for electrons, we've gotten down to sort of a couple of tens of MeV resolution so that we can see very fine features in, for instance, autoionizing states of argon and krypton when we've done sort of detailed look at that. But then when we do experiments with UV and VUV pulses in a pump probe measurement, our resolution is much poorer. And that's limited by sort of like the volume that we have in the focus and not so much by the camera. The ability to centroid the images and determine sort of sub-pixel resolution, the hit location, means that the camera is not the limiting point in the resolution, at least for our apparatus.
Next question is from Maurice. He says, if I understood right, you had an average event rate of 1 to 2 events per laser shot. If that is so, how sure are you that you do not have too many false electron-ion coincidences?
So that's a great question. I would say that for the coincidence work, no, we worked hard to get far fewer than 1 to 2 per shot. So Chuan did detailed calculations he was probably more worried than anybody else about the false coincidences. And I would say that for much of the coincidence work, we had less than 0.1. Or do you want to correct that, Chuan, if your recollection is different?
Yes. So the ion electron coincidence experiment was done with a way much lower acquisition rate. I think during that experiment, the average electron is about 0.4. Yeah. Oh, sorry, 0.2. Yeah, there's a factor of 2 due to the detection efficiency of the electrons. So at that time, I think our false coincidence rate is less than well, it's less than 1%, of course.
So you have a higher rate.
No. It's so that experiment. I mean, it's the same experiment. We just have the same aspects of the data. So I mean, if the electrons are like 0.2 per laser shot, then the ions will be less because the detection efficiency of the ions onto the MCP and phosphor is generally worse comparing to the electrons. So we actually have way less counts in terms of ions. Yep.
Question from Maurice. If he understands right, your average particle hit size on the timepics is about 50 pixels. And does that mean that the average diameter is the square root of 50, which is approximately 7 pixels?
You mean, I guess that's referring to the X and Y dimension? Yes, I'm referring to that. So basically, I'm picturing it as a square. Well, the hits are actually circular, but yeah, you know.
He's asking, why are the hits so wide? What kind of MCP pore sizes do you use?
So it's a good question. So we have recently purchased some imaging quality plates, which should decrease the size of the hits to some extent. But the estimates that you've made here are correct. So it's something like that. Although it does depend sort of quite sensitively on whether you're looking at ions or electrons, and whether you're using the part of the detector which is has high gain, low gain. So electron hits are typically larger, and ion hits are a little bit smaller. But we haven't sort of found where that's more of an issue is when you have overlapping hits. Because, for instance, if you're looking at sort of double ionization or events where you want 2 electrons, then a real problem is if the 2 electron hits overlap, because then it can be hard to sort of distinguish them, and you then get a false measurement that you measured a single electron, but there were actually 2. But we haven't found a sort of a substantial improvement in the resolution if you look at a single hit, depending on the size, although we haven't made a very detailed study of that. So the bottom line is that we hope to improve the size of the hits to some extent by using higher quality MCPs. Chuan, do you remember what the pore of the current MCPs is? Because you had placed the order, so maybe you remember that.
I think the current one is 25 microns, and the new ones that we have, it's 10 microns.
Shuai, he's asking about the time sequence of the ion lens. What is the time sequence of the ion lens? Quick switch from negative to positive.
So that's dictated by, so we use so these from DEI, we use these high voltage switches that provide timing switching less than 100 nanoseconds, kind of, you know, maybe a few tens, but probably under 100 nanoseconds. So what happens is that the electrons arrive within a few tens of nanoseconds after the laser pulse, and then we switch the voltage. Oh my, it's I'm having trouble with the fact that everything's back and forth in here, so putting my hands in the right ordering, I hope I get it right. But so electrons hit, then we switch the voltage, and then the ions arrive. And the voltage switching is accomplished sort of under 100 nanoseconds. So that's why I made this comment earlier about the protons. The main difficulty we have with protons is the voltage switching, not so much the resolution. But it's just that the protons move a not insignificant amount in that 100 nanosecond switching time and the time between the electron arrival and the switching, whereas the heavier ions don't really care, and so they're really unaffected by it.
Will that decrease the resolution of ion measure?
I'm not sure I totally understand the question, but I think certainly the problem is the switch. If there is problems with the switching, it's not so much of a statistical problem, it's more of a systematic issue, if I understand correctly. That is that there's sort of a systematic shift of the measurement from sort of reality with the protons during the switching, and so it's more of a systematic issue than it is sort of like just a resolution issue.
So I think, well, we can easily compare the scenario where we don't have electrons, so we only detect ions, and we compare that with the case where we switch the voltage, and clearly the easiest fragment to be influenced is hydrogen, because that has the smallest mass. The higher masses within that range of switching, they are not going to change much. Well, as far as I can tell, the hydrogen, the 3D distribution of it doesn't change that much. So I don't think it's going to affect our resolution significantly.
I believe we have one more question. Can one correlate the reading from the two cameras?
Yeah, maybe I can answer that questions. That's a good question. Actually, recently we developed the two cameras for the electron and ion coincident measurements. So indeed you can, because we developed, so we synchronize the two cameras to have the same clock. So that's you synchronize two cameras. Yeah. Then you obtain the data stream from two cameras, and they both have a timestamp. You can easily correlate them. So it's possible, and we already implement for one of projects in China. So thank you.
Thank you, Jingming. I can also see that we are almost at our 1 hour mark, but we have, I think, one more question. If you want to stay for discussions, feel free to do so. If you need to go, we understand. And thank you so much for joining. We wish you a lovely day. For the rest of the questions, we can continue for a couple more minutes. Let's see. There was one more question from Constant. For this voltage switch, are you limited in the gradient that you can apply, going from minus 2000 volt to plus 4000, for example, in 100 nanoseconds?
So I'm not sure what limits the time. I think there is a maximum slew rate for the DEIs. We certainly have found that the capacitance of cables and so on between the switch and the velocity map imaging matters. So we have tried to sort of minimize all of that. But I haven't sort of had a detailed study of the switching time versus or as a function of the voltage that you switch. We do frequently work, you know, sort of like at a couple of kilovolts. But we haven't done as high as 4,000 to 2,000, if I remember correctly. I think we go as high as like 3,500 or 3,000 at one point, right, Chuan?
Yeah, that's the spec of the high voltage switcher. They can only handle up to that. For the higher voltage ones, I think their response time is worse than 100 nanoseconds.
Any more questions from the audience? Let's see. Maybe Jinkming has a question. Yeah.
I want to ask.
Yeah.
Yeah.
There's one more question here from Theophanis. Yes, so I think you're asking a question that we also had the same experience with. That is, if I understand correctly, the question is reading, many years ago, we found that pulsing two open electrodes with the same pulsing unit and voltage divider resulted in image distortion. And I'm not sure that we saw image distortion, but we had the same problem. That is, we tried to say, oh, we're clever, we can just use a single voltage switch and we just use a divider. But it was very difficult to find a voltage divider that was fast enough and yet didn't draw too much current from the switch and the high voltage power supply. So we eventually ended up giving up on that approach because either we, the voltage divider was fast, but then it drew too much current and we went over current, or it was slow and didn't draw too much current, but then it was slow. So we just ended up going with the design where we have a separate switch for each plate, and we simply synchronize the switches, and that works very well. Of course, it's more expensive, but was more robust in our experience.
Single field VMI.
Yeah, Theophanis is also asking if this is single field VMI.
I mean, I'm not sure I understand exactly, but it's sort of dual slope in the sense that we have sort of 4 plates, 2 of them are at ground, and then we have sort of a high voltage, a 70%, and then ground. So it's, you can sort of think about it that you have a gradient like this and then another gradient like this. So it's, I would say, something like a dual slope. I think I did it incorrectly. So it's the voltages are typically for electrons, and we have something like 600 on the repeller plate, 450 volts on the intermediate plate, and then ground on the next plate. And so if they're spaced by equal distances, then the slope changes by something like a factor of 3.
All right, thank you very much. We have time for one last question from Jean Hendrik. What would be an upper limit of the repetition rate of the laser for these coincidence measurements? Would it be switching of the electrodes or the flight time of the ions?
So it's an interesting question. I have a colleague, Tom Allison, who was very interested in exactly this question because he has a 100 megahertz repetition rate laser. And in his case, he was limited by the time of flight, so that there would have been sort of, confusion between laser shots. I also think that there could be issues with the, and he was just thinking about electrons alone. If you have ions, then certainly the flight time of the ions are sort of microsecond type things, depending on your geometry, but I could imagine that would be an issue there. So we're hoping to sort of carry things out at about 100 kilohertz, but I'm not sure if how much higher one can go than that.
Yeah, I guess the DIs, the high voltage switchers, I think those can handle maybe 10 kilohertz in their spec. Well, we do have a limit, which is the arrival time of the ions, because the ions usually span for several microseconds. Let's say for 10 microseconds for safe. Then that limits our repetition rate to 100 kilohertz. Well, 100 kilohertz laser is not trivial anyway. So on the other hand, since you're talking about coincidence measurements, right? Coincidence is usually taking a lot of time because we have to suppress the detection rate to suppress the false coincidence. Well, we have recently been devoted into this covariance analysis, so that we increase the pressure so that we get way more events per laser shot. And then by doing this covariance analysis, we can get almost the same level of statistical fluctuation by boosting the acquisition time by almost a factor of 100. So that is a huge boost. So it's an interesting thing to do with the covariance, I would say.
Technically, the camera itself can accept 10 megahertz laser process trigger. So that's maximum.
Thank you again, all of you. We're really sorry, but our time has run out. And if there are any more questions, you can email us and we will come back to you. We would really appreciate your feedback on this seminar. So please take a minute to let us know how did you like it. And please stay until the end and you will be redirected to an online survey. Thank you again for joining and have a great day ahead.
Thank you. Thank you very much, Tom and Chuan. Yeah.
Okay, great to see you. Ciao.
Okay.
Bye, everyone.